Wonderful we are, so I shall take it from the very top. Okay, good morning, everybody, and welcome to the COVID-19 conversations. These conversations are brought to you every Thursday at 10 a.m. South African time by the African Alliance with the support from the South African Medical Research Council and the Department of Science and Innovation. They're also brought to you by the Vaccine Advocacy Resource Group, which is 100% funded by advocates, as well as the Community Constituency COVID-19 Front, the Treatment Action Campaign, and APHA. Colleagues, a reminder that the format of these calls, in the first 30 minutes, we will hear from our guests. In the last 30 minutes of the hour, we will engage in a question and answer discussion. Please feel free to put your questions, your comments, your thoughts in the chat box, and we will ensure that we get to as many of them as we can. Priority, however, will be given to questions that have been pre-submitted. Any questions that we don't get to during the hour, we have commitment from our guests that they will indeed work with us to get answers to those questions, which will be included in a post-call report. These reports, as well as all of our COVID-19 conversation recordings, will be available on the African Alliance website, which will be launching next week, and we welcome you to have a look at that website and engage with the vast amount of resources that will be loaded onto that platform. Colleagues, the HVTN is one of the world's largest HIV vaccine research networks. It conducts all phases of clinical trials, from evaluating experimental vaccines for safety and immunogenicity to testing vaccine efficacy. With the advent of COVID-19, the team that brought some of the world's largest HIV vaccine trials to South Africa are bringing their expertise, influence, and capacity to bear on a pandemic that has shifted our world. Simba Takuva, a medical epidemiologist, and Azwi Takalani, a public health medicine specialist, both work as medical monitors and safety physicians with the COVID-19 Prevention Network, CoVPN, and the HIV Vaccine Trials Network in the operations in Sub Saharan Africa. Leveraging on infectious disease expertise of the existing research networks and global partners, NIAID formed the CoVPN, whose mission is to conduct phase 3 efficacy trials for COVID-19 vaccines and monoclonal antibodies. Simba and Azwi join us today to describe to us the COVID-19 work in South Africa and the region, and build on one of our earlier webinars where Kagisho Baenyape and Dr. Jacqueline Odihambo introduced us to the work of the HVTN and the CoVPN, and gave us a teaser of what COVID-19 research is coming up in South Africa under the CoVPN banner. They will also describe the participant safety monitoring process in vaccine clinical trials. I'm sure you'll be aware participant safety has been a key topic on many of our minds, especially in the news cycles over the last few weeks. We've seen trials across the world having to pause and reflect and take guidance from the data safety monitoring boards in terms of participant safety and what that means for proceeding on various trials around COVID-19. So colleagues, without further ado, I will now hand over to both Simba and Azwi to share with us some of their reflections, thoughts, and give us a status update of the work that they do before we go into the Q&A session. Thank you again, everyone, for joining us. Thank you to Simba and Azwi for making the time to speak with us this morning in their very busy schedules. So over to you, colleagues.
Okay. Thank you, Tian, and thank you, Vivian, and the organization at large. Thank you for inviting us to talk on this platform. This presentation will be shared with my highly esteemed colleague, Azwi. I'll go first and she'll chip in later. So information sharing and engagement with civil society, the communities, is absolutely necessary for the success of scientific endeavors and innovations. So we really are thrilled to be on this platform, and we look forward to further continuous engagement. And just to also highlight that research is a partnership between different stakeholders, really. It's one whole village that goes through developing, be it a vaccine or a drug. So first, just as declarations, we have no potential, or should I say we have no conflicts of interest to declare. We're fully employed by the HVTN and the CoVPN. And as you know, the HVTN and CoVPN do partner with industry, academia, other organizations in clinical development of vaccines and antibody candidates. And maybe to start off, just introducing what I think is sort of like one of the highlights when you look at the whole GCP document, which is good clinical practice, the international ethical and scientific quality standard for the design, be it conduct, performance, be it monitoring, analysis, auditing, and reporting of clinical trials. So this is an international standard. And per GCP, it is very unequivocally clear that the rights, safety, and well-being of trial subjects or participants are the most important consideration and should prevail over interests of science and society. So as an outline, I'll lay the foundation by looking at the overall vaccine development process, then get into the bolts and the nuts of safety monitoring, and then Azwi will sort of take off from there. Then we'll also include some recent examples. I think Tian touched a bit about some pauses that have happened in the past few weeks. And we'll close by giving an overview of the ongoing HVTN and CoVPN studies. So first, let's look at an overview of the clinical trial process, or should I say, the vaccine clinical development process. So monitoring participant safety during clinical trials is a very critical component throughout the vaccine development process. So each step in a product's development cycle, be it the clinical testing and uses, provides information about the safety. So if you see this schema, it's showing the different phases of vaccine development, right from the preclinical phase before you actually go to humans. There is a preclinical phase. And then when you go into humans, you're looking at up to 4 phases. So we're showing 3 phases there. And if you look at those different phases, safety is emphasized. Safety is emphasized. So I would like to really highlight that establishment of safety for a vaccine starts way before that molecule is actually used in human trials. By the time you get to your first in human trials, which is like your phase 1, you sort of have like a good description of the safety of the molecule. And just again to highlight, clinical vaccine development is often described as consisting of 4 temporal sequential phases. So your phases 1 to 4. So that's after your lab studies and animal studies, you then move to the humans. So with your phase 1, this starts with the initial administration of an investigational vaccine into humans, whereas your phase 2 studies are conducted to explore therapeutic efficacy, really. And phase 3 studies are to demonstrate or confirm therapeutic benefit of the vaccine. And the phase 4 is what happens after vaccine approval. So we'll revisit these steps in the next few slides. And again, at each stage or at each phase, safety is emphasized. So a common question that has come up is the whole traditional model of vaccine development versus what we're currently seeing. So with your traditional model for vaccine development, each of those phases or stages are performed in sequence. So before you move on to phase 2, you need to have completed phase 1, and before you move on to phase 3, you need to have completed phase 2. So to accelerate COVID vaccine development, these steps are now being done in parallel. So just to highlight that all the usual safety and efficacy monitoring mechanisms do remain in place, such as adverse event or side effect surveillance, safety data monitoring, and long-term follow-up. So corners are not being cut. The time is shortened, but the processes still remain in place. The difference is that you're not doing it sequentially now, but you're doing some of these processes or conducting some of the steps in parallel. So if you think about it, additionally, beyond this clinical trial process development, you've also got process development and also manufacturing scale up. Yeah, so that also needs to be substantially accelerated by running all streams fully resourced in parallel. So moving on to maybe the preclinical safety bits. So I think it's really important to actually describe what happens before a molecule or a candidate vaccine is actually accepted to get into human clinical trials. So preclinical testing is the final deciding gate prior to clinical studies, that is your human studies. And only a few of those candidates that are tested in the preclinical phase actually advance to your first in human or to your phase 1 clinical trials. So if you look at some of the data from sources, to put it at like 7 to 12%, which is like roughly 1 in 10. So during this phase, the critical question that scientists and regulators seek to answer is whether that novel vaccine molecule is safe to be tested in humans. So really, this safety assessment starts at a very early age. So a lot of laboratory and animal testing happens to understand the pharmacology. So that is what the body does to the vaccine or what the vaccine does to the body. So that's your pharmacokinetics and dynamics. Also the safety and the toxicity profile So at the end of this phase or at the end of this preclinical stage, the most promising molecules are then selected for human testing. So this animal toxicity data or lab toxicity data is really essential so that you can be able to determine the safe starting dose and the dose range for your first in human studies, and also identify potential adverse effects that might be relevant to humans. So quite an important stage. And then the molecules that then qualify for human testing then also undergo the different phases or stages of the human clinical trial development process. And those that do not demonstrate acceptable safety or do not show an acceptable safety profile, they actually fall off and they do not make it into the human trials. So by the time we get to human trials, it means the safety of that molecule has actually been well described and understood. So you now then take it forward to humans to sort of like evaluate it even further. So the objective that is common across all those 4 phases of your human clinical trials is really the evaluation and establishment of safety. So across phase 1, phase 2, phase 3, phase 4, the common denominator is safety. So looking at phase 1, this is your first in human trial. So in as much as you are also evaluating biological activity in humans, you're also evaluating for safety and tolerability, building on your preclinical studies. And at this point in time, you also want to determine a maximum tolerated dose. I think if you remember, like, I think it's some Latin stuff I've read before, whereby they would say all substances really can be poisonous. What's important really is what dose are you giving, right? So if you look at your vaccines, your drugs, even though you establish that it is safe, like in your animal models, etc., or even in some of your humans, right? What is important is to actually then further describe or further understand, like, what is the maximum tolerated dose and at what dose should I stop increasing whatever I'm giving. So that is quite important. And then also at this stage, you're also obtaining information for the next phase, which is your phase 2. And then when you look at your phase 2, you're exploring efficacy in the indicated population, but at the same time, you're still evaluating safety. And now you're also looking at the dose and the response, like if I'm increasing the dose, am I getting a linear or an association with increase in efficacy, etc. And then you actually even determining the optimal dose that can be used safely and still achieving the desired therapeutic effect. And for instance, in vaccine, we'll be talking of immunogenicity. And at this phase, you're also obtaining information that will help you design your phase 3 trials. And then those compounds that then make it to phase 3, that's where you confirming efficacy in the indicated population. So with your phase 3, you're also identifying longer-term safety profiles. And at the same time, you're also looking at dose-response relationships. So these are trials that are usually multi-center, that have persons participating with varied characteristics, and looking at the vaccine trials that are currently ongoing, your phase 3s, you're looking at huge numbers in excess of 30,000 up to like 60,000. And the WHO is actually working on a collaborative effort where you're going to have like participant numbers into the hundreds of thousands. So you're also exploring, like, for instance, trying to understand the drug a bit more. And if you divide this phase 3 further, you can divide it into a phase 3B, where you like now extending what you understand in your phase 3 into special populations. Like, for instance, maybe the younger population, the pregnant population, maybe those with some form of impairments, etc. And after that, should the drug meet the rigorous minimum standards for licensure, you go into phase 4 studies. So even when a drug or a vaccine is licensed, the safety monitoring does not stop. So that's your post-marketing trials. And that's where you actually even start to identify your less common adverse events, because you now have much larger populations using the vaccines. And at this point, you also are able to refine the dosing recommendations further. So that was sort of like a nutshell of the whole clinical development process of vaccines, looking at like the preclinical stage up to your phase 4. And the common denominator when you look at all that is that safety is important. You have to establish it, you have to understand it, you have to describe it, and you have to drop off compounds that do not actually show acceptable safety. Now let's move on to the nuts and bolts of safety monitoring. So as you can see from this schema, I think what we are trying to portray with Azwi here is to sort of like illustrate that safety monitoring in a clinical trials involves a number of stakeholders and can be described as multi-layered. And all this is to monitor safety effectively, and also importantly to safeguard the volunteer, or to safeguard the subject, or to safeguard the participant. In the next few slides, we will briefly describe how each of the different stakeholders play a part in safety monitoring during a trial. And obviously, as you can see from the schema, if you look at like your extreme right and your extreme left, you've got the regulatory authorities and the ethics committees that are actually exercising oversight over this whole process. So we'll start by looking at the clinical research side staff and the investigators, then the study clinical safety staff, then the protocol safety review teams, and perhaps a word that you've had quite a lot the past few weeks, your data safety monitoring boards, or DSMBs. So starting at the clinical research site level, so this is where we also talk about the investigators. So if you look at your investigators, these are qualified physicians that are accountable for the clinical trial conduct, and they're working with a team of qualified varied personnel that are also supporting the success and the welfare of study participants. So they're protecting the rights, the safety, the welfare of the participants in the clinical trial. And the investigator really exerts study oversight and supervision. So they'll delegate trial-related duties to appropriately qualified persons. They make the trial-related medical decisions, oversee appropriate complete documentation and data quality, and also ensure communication of safety information is done in a timely manner to the regulatory authorities and the sponsors. I think later on, as we will sort of like take you through in terms of the minimum requirements in terms of reporting safety to the regulatory authorities. And all of this is teamwork that you see at the clinical research sites. And then moving on to the next level or the next layer. So this is after your clinical research site or investigator level. You now have the study-specific medical monitors and study-specific clinical safety staff. So this could be your safety physicians or your medical monitors or your clinical safety specialists. So at this level, you have persons that are fully employed, that are full-time, 24/7, performing blinded safety data reviews and making sure that they pick out any clinical events or any adverse events that may be of concern, and where there's need for clarification, those monitors or this team, they then get in contact with the clinical research sites and then sort of like perhaps go through the descriptions of the events, etc. And then if need be, this is then elevated to another layer, which we'll talk about in a bit. So they also maintain and respond to clinical issues 24/7. So just like in a hospital setting, you have doctors and nurses that are on call or night duty. They also do call rotas, having a 24/7 safety phone. And periodically, like with the VTN example, for instance, in some of the trials, weekly reports of aggregated safety data are generated, and then these weekly reports are then shared to the other stakeholders that are involved in safety monitoring. And then you actually have a collaborative, sort of like intervention from different stakeholders, different experts in looking at that safety data and making decisions that are in the interest of safety of participants. And then, for instance, even if sites have like clinical queries, like they have some medical issues that they'll need clarity on, that they'll want to discuss about, they contact this team. So this is a team that works 24 hours supporting the sites in terms of safety. So if you look at that diagram, there are some examples of that, of what that actually entails. So moving on to the next layer, I would like to hand over to Azwi, who's going to take us from your PSRT, which is your protocol safety review teams. And yeah, look at the other layers as well. So Azwi, I'd like to hand over to you. What I'm going to do, I'm going to stop screen sharing, and then maybe we'll switch to your screen. Thank you very much.
Okay, thank you, Simba. I'll just start sharing my screen. Okay. Can you see my screen?
Yes, Azwi. Thank you.
Okay, right. So taking off, are we on the right? On the right, I'm not sure which screen is sharing. Is it the Protocol Safety Review Committee?
No, so as we currently, you're on the cover slide on the first slide.
Okay. So,
now you're on Protocol Safety Review Team.
Okay, so just starting off with the protocol safety review team. This is a team that comprises various members with different levels of clinical and medical expertise, as well as a scientific background or laboratory expertise in terms of the product development. And on this slide, we're just showing a typical example of what a protocol safety review team, or what we call a PSRT, in our organization would comprise of. And the responsibility of this team is really to review safety data periodically. Like Simba mentioned, we review safety data maybe on a weekly basis as a collective. But however, if the safety team recognizes, or the investigators report an urgent matter, this is quickly escalated to the team to make an assessment on if the study can continue, or if there should be a pause while we review information and further action that needs to be taken. Okay, my slides are stuck. Okay, and the next layer is the Data Safety Monitoring Board, and this is sort of like oversight. These are people who are independent from the study or the protocol team, and they also have various subject matter experts, ranging from clinicians, biostatisticians, as well as patient advocates and ethicists. And essentially this board is unblinded. And the fact that they're unblinded means that they are able to see who is getting study product and who is getting placebo. And so when they do a safety review, they are able to determine if there are any trends or clusters that are more in the group that is getting safety, investigational product, and therefore they can also request for a pause in the trial for further review of the data to ensure that the trends that they are seeing that may be a bit skewed are not actually due to the investigational product. So they can make various decisions in terms of pausing the trial, and I'm going to get into an example of that after a few slides. Then we have the ethics committee, whose main role is really the custodian, or to safeguard the rights and the safety, as well as the well-being of all the trial subjects. And this committee usually comprises of 5 or more people, and as a collective, so no individual has to be an expert in all, but as a collective, they have to be able to comprehensively evaluate the science, the medical, the ethics, and the non-scientific aspects of the trial. And then they get to make a decision prior to the study being approved to say, is this appropriate or not? Should some modifications be made to the trial, or this sort of trial cannot be conducted as it is just not ethical? And on an ongoing basis, they also get to review the data, including safety data, and make an assessment on if the trial can continue. Then we have our regulatory authorities, and in the South African context, we have SAHPRA, which is the South African Health Products Regulatory Authority, and we have to report, there are some safety reporting requirements from them, and they require that the expedited reports for certain events, and expedited reports usually should be delivered to them, or they should be notified within a matter of days if any of the events under the expedited reports occur on your trial. And on a periodic basis, of which the most common is the 6-monthly progress reports, we also have to report all the safety events to SAHPRA, or our regulatory authority. And this includes everything in the expedited report and all other safety events that are not reported in a timely manner. They review this data, and they also make an assertion if the trial can continue and if the safety of the subjects on the trial is being covered. And then in addition to our regulatory authority, the sponsor may contract a research organization to assist them with monitoring and doing audits. And we may also have international partners or regulatory agencies who also require safety reporting and do audits. So just to take a step back and maybe define what an adverse event is, and then just to quickly go through the next few slides, explaining how we manage the various types of adverse events. So from the moment a trial participant is enrolled, and in vaccine trials or investigational product trials, enrollment means the first time you receive the investigational product. From that very moment until the study ends, then any untoward medical occurrence is deemed an adverse event. And this does not necessarily mean that it is related to the investigational product or the vaccine. It just means that, well, maybe you happen to get a cold and you did receive investigational product 5 months ago. So in terms of gathering the safety data, this too has to be reported. So then we have various types of adverse events that I'm just going to quickly go through. Unsolicited adverse events is all the adverse events. That's what I spoke about, to say all untoward medical occurrence from enrollment needs to be reported. And then the second one, and then we have subsets of this big group of all the adverse events that we are reporting that we want to focus on. And then we make rules on how they should be reported and how that information is managed. And usually additional information is required. And we pay more attention on these events. So we have solicited events, and this means that we particularly ask you, since you were last here with us, did the following occur? An example of this is like a reactogenicity event in vaccine trials, which is like a common vaccine related adverse event. So we're going to vaccinate you in the deltoid. You may have some swelling, you may have some redness. Now we're going to particularly ask you, did you, and for how long, and how big was the swelling, so that we can quantify those sort of reactions substantively. And when we, at the end of the study, we can be able to say 30% of people who received these products had these adverse events. Then we have adverse events that are serious in nature, and these are those that result in death, maybe life-threatening, require hospitalization, or the participant develops disability, or there is a congenital anomaly in a participant or a participant's partner who is pregnant. And the final one, which I have not put on the slide, is just to say a medically important event. And this category basically just gives leeway to the investigator to say, well, this event hasn't resulted in any of these 5, but I think it's serious. And therefore, this too should be reported in an expedited manner, and additional information will be required about that event so that we can really monitor and make a thorough assessment on what has occurred. Then we have adverse events of special interest, and this is just a subcategory of events that has scientific or medical concern to the vaccine. So we just say this product could, because we're trying to induce the immune system, so other immunogenic reactions such as could occur, and we make a long list. And should any of these occur, they also need to be reported in an expedited manner with additional information. Then the final one is a suspected unexpected serious adverse reaction. And essentially here, it's a serious adverse reaction, and we suspect that it's related to the vaccine. However, it's unexpected in the sense that in the trials, in the pre-phase trials, the phases that Simba described, so if maybe phase 2 or phase 1, these events were not noted in the literature, these compounds are not known to do this, so we did not list this as an adverse event in the investigator's brochure, but yeah, there is reason to think it's unexpected. And therefore, this also needs to be reported in an expedited manner with additional information. And so some of those events that I've just spoken about may trigger certain safety guns, which is something like a generic pause or a study stop rule. And essentially, when we pause a study, it's so that we can stop and review the data and elicit if there is a true safety concern that is related to the vaccine. And we can make a determination at the end of that pause that there's no evidence to link it to the vaccine. Maybe this participant or the subject had a hereditary condition and leave it at that. Or we could make a determination that maybe it's likely that it's related to the vaccine. And then we would have to take that back to ethics, regulatory, and everybody to say, can the study really be continued? Or other examples that are determined mostly by the DSMB is to say, if they see the unblinded data and they can see that the vaccination arm is really getting a lot of benefit compared to the non-vaccination arm, can we really continue for another year or two with the trial? Or should we stop and think about how we can make these products more accessible to more people in a cautious way without such a rigorous trialing process? So those are the various triggers for a safety safeguard. And I think in the vaccine, in the COVID vaccine example, we've seen those pauses happen in 2 of the trials, with the AstraZeneca trial, as well as the Janssen trial recently, and both have actually resumed giving investigational products or vaccinating, because when the evidence was reviewed, it showed that there was no evidence that the adverse events that occurred were linked to the vaccine. And when these things happen, I think we need to celebrate that actually the safety safeguards that were set in place are being adhered to. So there's no one accelerating and not paying attention to safety. And second of all, I think we'll see a whole lot more of these safety safeguard triggers in the COVID vaccines, because there is a lot of attention to safety, and therefore some of those thresholds for pausing have really been lowered to give us an opportunity to really ensure that safety is not being shortchanged. So just to wrap up, Tian, and I'm not sure if we still have any time left, but
thank you, Azwi. We are 7 minutes over time, but if we can wrap up, that would be great.
Okay, great. Thanks. So I'm not going to go through these trials, but I just wanted to highlight that we do have some CoVPN studies ongoing in South Africa, as well as we haven't abandoned our HIV work. All our trials that had already started and are ongoing, and we have one that also started recently. And I think that's the end. Thanks, Tian.
Wonderful. Thank you so much, Azwi and Simba. I wonder if I can ask us for our 2 guests to turn on their cameras if they're comfortable doing that, so we can engage with the participants. I do have some questions that we have received, and maybe we can just go straight into it. So Nita asks, please indicate when effectiveness of drugs is tested. What is the difference in efficacy and effectiveness in the phases of trials?
Azwi, I think I'll take that. Hi, Nita. Yeah, so that's an awesome question, right? So when you look at your clinical trials, generally you're looking at efficacy, and then that leads us to the question, what is the difference between efficacy and effectiveness, and when do we test for effectiveness? So efficacy really can be defined as the performance of your drug or vaccine under ideal and controlled circumstances. So that's in your clinical trial setting. Whereas with effectiveness, you're referring to its performance under real world conditions. Like for instance, you've demonstrated in those RCTs that the vaccine is actually efficacious. But now you also may need to show if it actually works in real world conditions. But however, the distinction between those 2 types of trials is some sort of a continuum rather than a dichotomy, or to say they are clear-cut black and white differences. So it may be unlikely, or it may be impossible to perform a pure efficacy study or a pure effectiveness study without some form of overlap. So as examples, for instance, in an efficacy study, you're looking at your control settings, like your clinical research sites that are well-resourced. And in terms of the participants that are participating, this is a homogeneous population that when you're selecting, you're applying strict inclusion and exclusion criteria. And your providers, these are well-trained providers with multiple layers of safety, as we've seen. But whereas in an effectiveness study, that's more or less like a field trial. So now you're saying, will this work in real-world clinical practice? And now you've got a heterogeneous population where you've got everyone really who would like to receive the vaccine, and they do not have any exclusions or any concerns receiving that vaccine. And you have your usual service providers in the healthcare system. And last but not least, you could think of this like in terms of the malaria vaccine, the RTSS vaccine. So clinical trials were concluded and it was shown to be efficacious. But what is currently happening now in Kenya, Malawi, and Ghana, there are large field studies to sort of like inform broader implementation. And those are sort of like effectiveness trials. I hope that answers your question.
Great, thanks so much, Simba. So we can go straight into the next question. I think this one relates to the DSMB. It says, would the DSMB, for example, have on it anyone who has a vested interest in the outcome of the trial or materially benefit from having access to trial data in their professional lives? I hope the question came across clear. So on the DSMB, would you find people who have a vested interest in the data being generated by their trials in their normal course of work?
Okay, so I'll take that one. Thanks. I didn't get who asked the question, but I think the general rule is that it should not be someone with a vested interest, right, in the trial actually succeeding. So having said that, I think one of your points was, would they benefit in their career? And yeah. I think that becomes a hard distinction. If you think about it, if you are all medical practitioners, you know, we would all really benefit from the burden of disease, of certain infectious diseases, you know, being really low. So in that sort of like removed roundabout way, then you could eventually benefit, right? But it should not be someone who can directly benefit from the outcome of the trial. It should be an independent review. I think that's the one word that we should have added on the slide to say it's independent subject matter experts who then get to review this data, review the data for us. So while in a DSMB, these are big phase trials, you need to be independent. But in the small phases trials where you are in the organization, you're thinking, we're moving from preclinical to clinical, you may have an internal SMB that may look at the data. And at that phase, it's not making determinations about large populations. But essentially a DSM board should have independent subject matter experts. Thank you.
Thanks for that. There's a question here on communicating adverse events. I remember a couple of years ago, I think it could even perhaps be a decade ago, when I was sitting in a conference and the researcher put up the adverse event slide almost towards the end of the presentation. And they were never, quite clearly described in, simple language, but we had seen the images. And it just struck us, I was sitting in a group of activists, and it just struck us how it almost came across as lightly communicated the adverse events were. So I wanted to hear from you, in your experience, what could we be doing better or differently when we communicate around adverse events so that it perhaps does not come across to the general public as minimizing something that might be a rash or headaches, etc., etc. So could you very briefly just describe that continuum? I know you have a whole slide on it, but my question is really focused on the communication of adverse events. Do you have any thoughts to share on how adverse events are communicated and their potential impact on trial participation and getting communities excited and interested about participating in trials?
Thanks, Tian. So Azwi, I can go first, and if you've got ads, you can jump in. Yeah, so that's a great question as well. So when one thinks of it again, you think of it as a continuum, right? Right from the start, from the time that you're drawing up the protocol, it needs to be clear that adverse events, like for instance, your phase 3, your phase 2, your phase 1, safety is a priority. So if you look at all protocols that we work in, beyond efficacy, one of the key primary objectives is to establish and also to test safety. And then with that protocol, it then moves on to your informed consent document. So again, with your ICF, this is a very important document. Safety, adverse events, expected adverse events, be it solicited or unsolicited, this needs to be laid out clearly in the ICF so that participants can be able to have a good sense of the type of side effects or adverse events that are involved. And then when it comes to communication with the general public, so that's outside the clinical trial setting, beyond your subjects, there is an importance with regards to improved transparency. There is an importance with regards to improved transparency. And I think we've seen that in the past few weeks where there have been maybe some adverse events that have resulted in trials being paused. There's been transparency to sort of like report this to the general public, but at the same time, trying to balance the rights, the privacy, confidentiality of the participants that are involved. But yeah, there's room to always do better with regards to that. I don't know, Azwi, if you can maybe add a few points to that.
Yeah, I think you covered it really well, Simba, but maybe just to add that for the trial participants, that's why I think it's important that the ethics committee also reviews the informed consent and the informed consenting procedure. So if you think about it, we're doing global multi-center studies, and an adverse event that may seem really minor in one setting may have a cultural bearing in another setting or more important. And so when we do our informed consent process, when we try to put as many adverse events there, but we also encourage the staff that will be taking the participants through the informed consent procedure to highlight those adverse events as far as possible. And the ethics committee can also come back and say, well, we think that in this context, you need to really communicate these adverse events more to the participants or to the community prior to you even starting with the enrollment of participants. So those are some of the communication mechanisms that we really try to use or that are in place to ensure that adverse events are communicated well to the trial participants. And then, like Simba said, thereafter, it becomes an issue of the people who will then, this is post-trial, then it's the clinicians. Are you explaining these things well to the participants before, post-licensure, is the community aware? And I think with time, it's becoming more apparent that lack of adherence, even trial loss to follow up on trials, is because participants in the community were not aware of certain adverse events. And when they happen, they become shocked and they pull back. So the pressure is increasing to be more transparent and to have better and better communication strategies of adverse events.
Yeah, absolutely. Thanks for that. The next question I have here is, could either of you describe briefly what discussion happens with researchers in terms of access? So are researchers having access discussions when trials are designed, conceptualized? Is that forefront of, in your mind, the access issue, the eventual access of the product? So could you very briefly just describe researchers' role in the access conversation?
Okay, thank you for that question. I'll have a go first. For instance, by the time you get to your late phase studies, your phase 3 studies, There are lots of investments that have come in, and at that time, conversations around access usually are already fully established. Like, you have to prepare for success, right? Should you succeed in coming up with a vaccine that actually works, what happens after that? And also, you have to prepare for failure. So we're thinking of a hypothetical situation where you're preparing for success, and that means talking about access. How do you roll out the vaccine? Who gets the vaccine, etc.? So one of the things really to highlight is that the concerns that usually come out is mainly bent on the fact that you have your less resourced nations or countries that probably are unable to maybe fund some of the studies or even buy some of those vaccines.
Yeah.
But thankfully, due to the current global initiatives, be it in HIV, be it like in COVID-19, there are models that are working well, like your public-private product development partnerships, where you have well-resourced funders or well-resourced countries that throw in resources or put in resources into the kitty bag, so that there can be equitable access for all. An example would be an organization like, for instance, the Gavi Alliance. And recently, I'm sure you've heard of this, the WHO has been putting together a facility with different organizations, which is known as, I think it's called COVAX, Access to COVID-19. So it's a framework whereby the WHO has developed that sort of like guides and gives guidelines for equitable and affordable access to safe and effective vaccines. So the whole aim of this framework is that at the end of 2021, should there be a vaccine, there should be enough resources to actually deliver beyond 2 billion vaccine doses globally, regardless of how rich your nation is or how poor your nation is. So vaccine allocation will also be driven by public health needs for priority groups, which might actually represent close to half of the population.
Yeah.
So yeah, there is a structured conversation going on, and there's a lot of effort behind the scenes that's going on, that's setting the stage for rollout should there be success.
Great. If I can just ask, for planning purposes, we have quite a lot of questions that have come through both on email and some in chat. Do you both have a hard stop at 11, or could we perhaps continue until 1:15?
Okay, maybe we can make, I have a hard stop, but maybe we can, yeah, let's make it up to 1:15. I think it's fair. I think it's fair to do that.
Thank you. So a question on the chat from Ntando is, speaking of varying cultural settings, how well is the field doing in innovating informed consent processes? In fact, does Simba and Azwi think there are opportunities for improvement on how we currently do informed consent?
Yeah, that's a great question as well. So Azwi, I think I can go first and you can add on. And then I think we have Kagiso as well. So Kagiso, feel free to jump in. That's a great question, Tando. And I think as the times progress, we also have to be innovative and also have to sort of like adapt and align to the different environments that we work in. So perhaps what immediately comes to my mind is the way the informed consent process has sort of like adapted to the current status quo, which is like a pandemic like COVID, lockdowns where persons cannot then travel reliably, etc. So there have been some innovations, like for instance, being able to go through informed consent, not in a face-to-face environment, but perhaps like remotely, whereby you can be in touch with the participant and then take them through the informed consent process. And then they are able to, for instance, maybe electronically sign that. So that's a kind of innovation that I can think of that's sort of like aligned to the current status quo. But then, of course, there are cultural caveats that you do raise, and I'm sure there may be more examples to that. Azwi or Kagisho, do you want to add a bit more to that?
Let's just see. Kagisho still here with us?
Kagisho, are you here? Okay. So
yes, I'm here.
Okay, I'll give Kagisho a chance first.
Great.
Thank you for unmuting me.
Hi,
So one of the things that are done in terms of the ICF is that, like Simba said, there are different ways that we are trying, but obviously like issues of new, where ICF is on devices, but also we need to give participants a chance to, like, for example, take the information home to consult with the doctor. But the question that I ask, it's important because we need to have also to have new ways, because we can't be doing things as we have been doing before. And one of the things that sites are doing, that, for example, they will have flip charts, or the whole ICF on a flip chart to discuss with the participant, besides giving the participant the document to take it home. They will have like videos where ICF is narrated on a video, but still the participant has to take, will still have to sign it on a paper form. But what is important is that participant is not coerced to sign the ICF at the same time. If you want to take it home, you can take it home. But I think this time, the times that we are living in now are really pushing us to innovate new ways of doing ICF than what we normally used to do. And thanks, Ntando, for asking that question. I think it really makes us to think to what are other ways that we can do. But these new studies are trying to check if we can't use those devices for ICF, for example.
Yeah, great. Thanks so much for that. Kagisho, colleagues, I wonder if we can power through to the next question. It seems like a very straightforward question, but it also feels like it's quite deep. Antibodies or vaccines for COVID-19, which shows more promise?
Okay, I'll go first, and then I'll give Simba an opportunity to add. I think the way we are thinking about COVID, it's a pandemic. Millions of people all around the globe are affected. And so the target is to have at the end probably a multi-pronged strategy. So having said that, both antibodies and vaccines are showing promise, but the question should really be more for which target groups do we think one will work for the other, and in terms of that, where are we showing more promise? So for example, looking at giving antibodies to maybe more the elderly, is that showing like it's going to show more promise of really preventing severe disease? But if we think about a younger population cohort, will the benefit of those antibodies be sustained enough for a long enough period for us to think that this is a good preventive strategy? So I think the question is, like you're saying, Tian, it's straightforward but deep, because it needs to be thought about in different layers, but which just so happens to be the answer of the approach towards COVID prevention strategies, that they should be multi-pronged, targeting different population parts to achieve different results. So both show promise at different levels. Simba, do you want to come in there?
Azwi, I couldn't add more really. I think you've captured it very well. Maybe I could just add like an example in another therapeutic area. If you think of family planning, your ideal scenario is you want to have a toolkit of interventions, right? So like for instance, in family planning, we have the pill, and the pill really may not be suited for everyone. I might be forgetful, so maybe taking a pill every day might not be the best. I might need something that is long-term, like an injection that I have every 3 months. Then someone might want a copper IUD or something like that. Yeah, so at the end of the day, you're developing really a toolkit or a box that has a lot of those different interventions that might be appropriate for different people in your population, just as we really articulated it. Thanks.
You know, absolutely. I think as much as we advocate for this fabled basket of prevention options in HIV, I think the same applies to research as well. I have actually 2 more questions, so I might be able to wrap this up and allow people to fully participate. So this question speaks about the halts and the pauses that we've seen globally of a range of COVID trials. Have you seen this impacting at all your ability to recruit participants for your trial? Have people shown some hesitance regarding being enrolled in your trials because of this? And that's a question from Anna in Harare.
So maybe I can go first. So from what we hear with colleagues that have already started those trials or colleagues that have had holds and pauses already, I think what really happens is you tend to get more questions. And this is where the issue of transparency really becomes very, very important. So what is really important is to then inform your volunteers, inform your subjects, inform your population, inform your collaborators. If there is transparency and the information is out there, then people are able to take in the information, comprehend, ask further questions if there is need. And usually that actually even instills more confidence in the clinical trial system to say those safeguards are actually working and are actually in action. So it comes back to communication and transparency. Yeah.
Great, thanks for that. The last question that I have here is, what is your take on how different community engagement has been in COVID research compared to HIV prevention trials? Has there been a difference?
Azwi Kagisho, do you want to take that one first?
Let's give Kagisho a chance and then I'll add on. Kagisho.
Okay.
Oh, thank you for unmuting me. Yes, I would say there has been a difference in terms of how COVID-19 community engagement, for example, has been done compared to HIV, for example, HIV vaccines or HIV Vaccine. So one, because of the challenges that we have faced with COVID, and the ability of people to meet, access to some of these things. But we also want to appreciate that it has not been easy, but things had to change. One of the things was that the issue of time was an issue for the engagement to be done as we would have wanted to be compared to HIV vaccine. But what we are happy about and encouraged about is that we have managed to speak to people on the ground and engage with communities, and our sites have given us a good rapport in terms of how their communities have engaged with them. So like we we said previously in one of the questions that you asked, Tian, that COVID is actually pushing us to look for new avenues on how we can communicate and improve on our engagement, and which is also what GPP has been teaching us, your broader stakeholder engagement. And this is what we also, for example, in every conversation that we have with community educators, is to ask, how broad are we moving? Are we moving beyond our traditional engagement areas? So it is what it is, and it is moving us to change, and I believe it's changing for the better and in the right direction to ensure that we continue to engage communities broader. And broader means it's not necessarily if we have engaged different people today, tomorrow you would engage a different stakeholder, but it says we would continue to engage different communities that in some instances we have never engaged, or stakeholders that we have never engaged with before. So what is nice about this process is that it has pushed us to actually talk to groups or people that we have never perhaps had engagement with in previous engagements.
Yeah.
Yeah.
Absolutely. Thank you for that, Kagisho. I mean, also just to reflect on the previous point around hesitancy, etc. It's certainly a challenge, and just to put that out there, so hopefully Simba, Azwi, and Kagisho can also share the sentiment in your circles of influence. Is that it's been a long time challenge with civil society and advocates in the way we communicate with researchers and scientists. One of the trials that was paused in South Africa, when we actually had participants who were coming to us saying, we've just heard from the BBC that the trial that I am a participant of was paused. It's the first time I'm hearing it. And so when we went back to the trial said, no, we've communicated to all participants. But we actually had participants who were saying, nothing has happened. We've not received the SMS. We've not received the phone call. We're clueless. We're hearing about this on the internet. And so it's about that balance of the trial having to protect that level of confidentiality, but also just being open in terms of where systems are weak or systems need to be strengthening or where systems are failing. Because the reality very often that we see is polar opposite to what the researchers and scientists are telling us. But I'm really happy that we've got an opportunity to have you all in this space. And thank you for allowing us to go 7 minutes over time. And thank you to all the participants. Colleagues, Simba Takuva, a medical epidemiologist, and Azwi Takalani, a public health medicine specialist, both work as medical monitors and safety physicians with the COVID-19 Prevention Network and the HIV Vaccine Trials Network. We also heard from Kagisho Baenyape, who has a background in nursing and primarily as a chronic nurse doing education in medical wards at Klerksdorp and Sepong Hospital Complex. Currently, he's the Community Engagement Project Manager for HVTN and CoVPN Africa region. Simba, Azwi, Kagisho, thank you so much for spending some of your Thursday morning with us. We hope this is the beginning of an ongoing conversation. We've been allies and comrades for many years, and so we hope this builds on that relationships and takes us further along the road to transparency, accountability, and meaningful engagement. So thank you for your time, and thank you to all of the participants for joining us. The month of November brings exciting things. We speak to South Africa's 4 leading political parties on what they are doing for their members in the COVID-19 space and what their experience has been engaging with researchers and scientists working on solutions to COVID. So thank you again to everyone and have a great Thursday morning, and we look forward to speaking to you next week.
Thank you very much. Appreciate the opportunity.
Thank you, Tian. Thank you, everyone. Bye.